It's hard to believe. We've completed 2 introductory lessons. You should now be up to speed and in the coming lesson, ready to tackle the world of viruses. In Lesson 2, we first reviewed the chemistry of polymerization, and then we learned about DNA, Chargaff's rules, how Avery discovered that DNA is the transforming substance, and ultimately, how the double helix simply makes sense. Here, the following might help to summarize some of the main points. Polymerization is linear and directional. Water is produced through condensation. The monomers of DNA are A, T, G, and C, and are called deoxynucleotides. Nucleotides link always 5' to 3'. That is, by adding the 5' end of the incoming monomer to the 3' end of the polymer being extended. Replication proceeds through AT and GC base pairing. RNA is the working copy of DNA and contains A, G, U, and C, the ribonucleotides. Proteins are amino acid polymers. The 20 amino acids can be polar or charged or hydrophobic. The genetic code consists of 3-letter codons. Ribosomes recognize the reading frame and translate messenger RNA to produce proteins. The following concept maps further explain the interrelationships between the terms we've just learned. Nucleotides contain 5 carbon sugars, phosphate, and nitrogenous bases. Ribose and deoxyribose are the sugars that make up DNA and RNA. Deoxyribose is the sugar for DNA, and it can bind thymine, cytosine, adenine, and guanine. Ribose is the sugar of RNA, and it can bind adenine, guanine, cytosine, and uracil. DNA and RNA form polymers. RNA is a single-stranded polymer, while DNA is a double-stranded polymer. The bases can base pair. G base pairs to C, A to T. Polymerization links monomers via a process of condensation, which produces water and polymers. Depolymerization takes polymers and via hydrolysis at the expense of water generates monomers. DNA is transcribed into RNA. RNA can translate via the genetic code to create proteins, which are polymers of amino acids. The genetic code is made of codons, and each codon contains 3 bases. Amino acids contain a central carbon, carbon alpha. Carbon alpha is linked to a carboxyl group, COOH, and an amine group. The 4th valency is a residue, and the residues can be hydrophobic or polar. The polar can be charged or non-charged, and the hydrophobic can be aromatic or just hydrocarbons. The amino acids link together via peptide bonds and ultimately produce polypeptide chains and proteins.